Welcome to Section 4 of General Principles. Throughout this section, I will be discussing gradients and action potentials. Let's get started. Before potentials, we need to build some foundational knowledge by understanding ion channels, electrochemical gradients, and membrane potentials. An ion channel is a protein that spans the cellular membrane, and it's selective because it only allows the passage of anions or cations. For example, a selective anion channel will only allow anions like chloride to enter the cell. And at the same time, it will prevent the passage of cations or other solutes such as large molecules. Ion channels can be open or closed, and this is controlled by what physiologists refer to as gates. The gate to an ion channel is simply a portion of the protein that changes shape under different circumstances and can prevent the passage of ions when it's in this particular configuration. If an ion channel is very permeable, it means the protein spends more time in a configuration that allows for the passage of the ion. If an ion channel is not very permeable, it means the protein spends less time in a configuration that allows for the passage of the ion. So if we have increased permeability, it means increased passage of the ion. On the other hand, if there is decreased permeability, then there's decreased passage of the ion. For step one, it's important to be familiar with two types of ion channels: voltage-gated and ligand-gated. A voltage-gated ion channel is opened or closed due to changes in voltage, which we'll talk more about in a second. A ligand-gated ion channel is opened or closed by the binding of hormones, neurotransmitters, or other ligands. Okay, with this basic knowledge, let's discuss chemical and electrical gradients. There are two types of gradients that form as a result of the permeability of ion channels: chemical and electrical gradients. First, I'll discuss chemical gradients and then electrical gradients. A chemical gradient is formed due to the difference in ion concentration across a permeable membrane, which produces a driving force in which ions diffuse from the high concentration compartment to the low concentration compartment. It's important to know that the size of the gradient is dependent on changes in concentration. This can be confusing, so let's take a second to draw this out. So we'll say this is a cell. We'll call cell A, and this will represent a permeable membrane. For simplicity, let's say the intracellular space, which we'll label right here with an I, is empty, and the extracellular space, which we'll label right here with an E, contains sodium and chloride. In this case, let's assume the membrane contains ion channels that are permeable to sodium. So sodium can cross the membrane, but chloride cannot. We'll represent that here by this arrow. Because the concentration of sodium in the intracellular space is low, and the concentration of sodium in the extracellular space is high, so low sodium, high sodium, a chemical gradient has been formed, which means sodium will naturally move through the ion channel from the extracellular space into the intracellular space. So we will draw 3 sodium molecules in the intracellular space and erase 3 from the extracellular space. If the chemical gradient was the only force acting on this system, then eventually sodium would equilibrate between the compartments, so we'd expect to see 3 molecules of sodium in the intracellular space and 3 molecules of sodium in the extracellular space, which is what I've drawn here. As I'll explain in a second, however, there is an electrical force that prevents the concentration from equalizing. Let's redraw this same example, but now assume the membrane contains ion channels that are permeable to chloride. So we'll say this is example B. Here's a permeable membrane, the intracellular space and the extracellular space. So in this example, we'll say chloride can cross the membrane, but sodium cannot. Because the concentration of chloride in the intracellular space is low, and the concentration of chloride in the extracellular space is high, a chemical gradient has been formed, which means chloride will naturally move through the ion channel from the extracellular space into the intracellular space. So again, we'll erase 3 chloride molecules here in the extracellular space and show that these 3 have been moved into the intracellular space. It's important to know that the driving force I just described is also sometimes referred to as diffusion potential. So this can be called a chemical gradient or diffusion potential. Okay, now let's discuss electrical gradients. Gradient is formed due to the difference in charge across a membrane. Difference in charge across a membrane. This charge difference produces a driving force in which ions are attracted or repelled to the different compartments depending on the size and sign of the gradient. Let's draw this out. Again, let's say this represents cell A with a permeable membrane. Again, this will represent the intracellular compartment, and this will represent the extracellular compartment. We'll say the extracellular compartment contains sodium and chloride. In this case, let's assume the membrane contains ion channels that are permeable to sodium. We'll represent that here with this arrow. Because the concentration of sodium in the intracellular space is low, and the concentration of sodium in the extracellular space is high, a chemical gradient has been formed once again, which means sodium will naturally move through the ion channel from the extracellular space into the intracellular space. This time we'll show one sodium molecule in the intracellular space and erase it from the extracellular space. As one molecule of sodium moves from the extracellular space and enters the intracellular space, you'll notice a difference in charge begins to occur across the membrane. Because the extracellular space loses a positively charged sodium molecule, this compartment becomes more negative. So we'll draw in negative signs right here. Because the intracellular space gains a positively charged sodium molecule, this compartment becomes more positive. So we'll draw positive signs on this side of the compartment. This charge difference produces an electrical gradient or force that begins to counteract the chemical gradient. This makes sense. The positively charged intracellular space will begin to repel additional positively charged sodium molecules because two positives repel each other. Additionally, the negatively charged extracellular space will attract the positively charged sodium molecules that are attempting to leave the compartment because a negative and positive attract each other. In other words, the chemical gradient forces sodium to go this way, and the electrical gradient forces sodium to go this way. Electrical gradient and chemical gradient. So you can see that there are two opposing forces that can cause ions to move to the different compartments. Forces caused by the chemical gradient and the electrical gradient. When these forces become exactly equal and opposite to each other, this is referred to as the equilibrium potential. The equilibrium potential for a particular ion can be calculated by using the Nernst equation. The Nernst equation isn't necessarily a high-yield equation to memorize. It's more important to understand the equation conceptually, which is why I'm explaining it now. So here is the equation, and E is the equilibrium potential measured in millivolts. Z is the charge of the ion. For example, if we were calculating the equilibrium potential of sodium, then we'd use +1 because sodium has a +1 charge. Likewise, if we were calculating the equilibrium potential of chloride, then we'd use -1, or if we were calculating calcium, then we'd use +2. R, T, and F are constants that can be ignored for step 1. And Ci is the intracellular concentration of the ion, and Ce is the extracellular concentration of the ion. Let's do an example. So we've been given parts of the Nernst equation and we're asked to solve for the equilibrium potential of sodium. Recall that the full equation is E equals -2.3 RT over ZF log base 10 Ci over Ce. We can see that all of the constants, so -2.3 RT over F, this whole part right here, was combined into a single value of -65 millivolts. And then Z is still right here. We also know that we're calculating for sodium, so Z would be +1. Finally, we can plug in the intracellular and extracellular concentrations, which we've been given right here, which is 10 over 100, or 0.1. It's very unlikely that you'll have to do log base 10 calculations on step 1, so I'll just tell you log base 10 times 0.1 is -1. So this entire part of the equation is -1, and this part of the equation is -65. If we plug all of this in, we get E equals -65 times -1, which is +65. Again, this equation is unlikely to show up on step 1, but I do think understanding what this means conceptually is very helpful in understanding equilibrium potentials and action potentials. So let's take a step back and explain this. To help illustrate this, let's draw out a cell. We'll say this is the extracellular space, and this is the intracellular space. And then, as I've drawn here, this represents the cell membrane. In this example, we're using a hypothetical situation and assuming that the cell is only permeable to sodium. So we'll draw a sodium ion channel right here. We know that the extracellular concentration of sodium is 100 millimoles. So sodium equals 100 millimoles. We also know that the intracellular concentration of sodium is 10 millimoles. Let's also assume there are other ions outside of the cell, such as chloride, but the cell is impermeable to these. As I showed earlier, the positively charged sodium leaving the extracellular space will cause it to become more negative. So here I'll draw some sodium molecules. We'll show sodium leaving the extracellular space and entering the intracellular space. So as this happens, the extracellular space begins to become more negatively charged because it's losing a positively charged sodium molecule. And on the other hand, the intracellular space begins to become more positively charged because it's gaining a positively charged sodium molecule. The charge difference will create an electrical gradient, which I'll draw right here, that prevents the additional influx of sodium into the cell. The example we just went through with the Nernst equation asked the question, how strong does the electrical gradient have to be in order to prevent additional sodium from entering the cell? Let me repeat that because I think it's incredibly important. The example we just went through on the previous slide asked the question, how strong does the electrical gradient have to be in order to prevent additional sodium from entering the cell? From our calculation, we found that the inside of the cell has to be positive and the voltage across the membrane must be 65 millivolts. In other words, at positive 65 millivolts, the electrical force is strong enough to prevent the influx of additional sodium. Okay, let's go back. So again, the Nernst equation allows us to calculate this value if we know the intracellular and extracellular concentrations of the ion, in this case, sodium. So if you're asked what the equilibrium potential of an ion is, in this case, sodium, then the test writer is really asking you the question, how strong does the electrical gradient have to be in order to prevent additional ions from entering the cell? Okay, we just went through a hypothetical example of a cell that's only permeable to sodium and determined the equilibrium potential. In reality, the cell membrane is permeable to many ions, so many equilibrium potentials have to be considered. This brings us to the next topic, resting membrane potentials. A resting membrane potential is similar to an equilibrium potential, but it's determined in part by all of the various ions trying to reach their individual equilibrium potentials. So multiple ions attempting to reach their individual equilibrium potentials. In the previous example, the cell was only permeable to sodium, which means the equilibrium potential of sodium would have been identical to the resting membrane potential. In this case, the equilibrium potential and the resting membrane potential of the cell would have been positive 65 millivolts. In actuality, the resting membrane potential is different from positive 65 millivolts. For example, in nerve tissue, the resting membrane potential is negative 70 millivolts. So why is the resting membrane potential of the cell different from the equilibrium potential of sodium? This is primarily a result of potassium leak channels. Let's draw this out. This will represent the extracellular space, and this will represent the intracellular space. And this will be the cell membrane. Recall that cells have a sodium-potassium ATPase pump. This pumps 3 sodium molecules out of the cell in exchange for 2 potassium molecules that enter the cell. This pump is responsible for establishing the concentration gradient. In other words, it actively pumps sodium into the extracellular space, so the extracellular concentration of sodium is high, so high sodium, and the intracellular concentration of sodium is low. It also results in a high concentration of potassium in the intracellular space and a low concentration of potassium in the extracellular space. Some students attribute the sodium-potassium ATPase as the primary reason for the establishment of the negative 70 millivolt resting membrane potential. They think, oh, 3 sodium molecules leave the cell and 2 potassium molecules enter the cell, so eventually the inside of the cell will lose more positive charges and will have a negative resting membrane potential. However, this is not accurate. Let me say that again. The sodium-potassium ATPase is not primarily responsible for the establishment of the resting membrane potential. Studies have been done that alter the sodium-potassium pump such that 2 sodium molecules leave the cell and 2 potassium molecules enter the cell. And in these studies, the alterations in the pump did not significantly alter the resting membrane potential. In other words, when the sodium-potassium pump transports ions in a 2-to-2 ratio, the resting membrane potential still remains around negative 70 millivolts. So what is responsible for the resting membrane potential of negative 70 millivolts? To answer this, we need to remember that there are also sodium and potassium leak channels, which allow sodium and potassium to diffuse down their concentration gradients. So this will represent a sodium leak channel, which allows sodium to enter the cell, and this will represent a potassium leak channel, which allows potassium to leave the cell. It's important to know that sodium leak channels are not very permeable to sodium, so low permeability. In other words, only a small amount of sodium can diffuse down its concentration gradient. On the other hand, the potassium leak channel is very permeable to potassium, so increased permeability. This means more potassium leaves the cell compared to the amount of sodium that enters the cell. So you can see that over time, the loss of potassium will cause the cell to be more negative, which is the primary reason the cell has a resting membrane potential around negative 70 millivolts. With this foundation, we can now discuss action potentials. Before we jump into the steps of an action potential, let's define some important terms. Inward current is the movement of positively charged molecules into the cell. Outward current is the movement of positively charged molecules out of the cell. Depolarization is when the membrane potential becomes more positive. Most commonly, an inward current of sodium will result in depolarization. Hyperpolarization is when the membrane potential becomes more negative. Threshold is the membrane potential at which an action potential must occur. Some of these definitions can be confusing, so I'll be sure to explain them as we go through an illustrated example of an action potential. Okay, so let's draw this out. Let's say this is a cell, this is the intracellular space, and this is the extracellular space. The potential difference across the membrane can be measured with a voltmeter, which is simply done by placing one electrode in the intracellular space, so draw that here, and another electrode in the extracellular space, like this. Also draw in another voltmeter right here. The voltmeter simply measures the extracellular voltage and subtracts it from the intracellular voltage. So the intracellular voltage minus the extracellular voltage. This value gives us the potential difference, and under normal circumstances this would give us the resting membrane potential, which is negative 70 millivolts in neurons. I'll also draw a graph showing the change in the intracellular voltage over time. So this dotted line right here will represent the resting membrane potential. This is a nerve, which is about negative 70 millivolts. Now let's draw a ligand-gated sodium channel in the cell. Recall that ligand-gated channels open when hormones, neurotransmitters, or other ligands bind to the channel. In this case, let's say a neurotransmitter binds to the channel, which causes the gate to open and allows sodium to enter the cell. Sodium enters the cell. This inward current, or positively charged sodium molecule entering the cell, will cause the other positive charges in the cell to begin to repel each other. So positive charge enters the cell, and it causes other positive charges to move. As you can see, positive charges begin to scatter throughout the cell, and a ripple effect is observed near the voltmeter. In this case, the red voltmeter detects the increase in positive charges, so the voltage on the inside of the cell begins to increase. We'll represent that here on the graph with the red line. You'll notice that the blue voltmeter is slightly further away from the ligand gated channel, so the increase in positive charges reaching this area isn't as significant as the red voltmeter. So we'll draw that here, but it won't be as significant. From the graph, we can see that both voltmeters were able to detect the cellular depolarization that occurred after the neurotransmitter bound the ligand gated sodium channel. Let's take this one step further. Again, this is the cell, this is the intracellular compartment, and this is the extracellular compartment. And again, we'll draw two voltmeters, a red voltmeter and a blue voltmeter, and this will represent our graph. This will be the resting membrane potential, and -40 will represent threshold. These lines here will represent the voltmeters that could detect changes in voltage. Here is the ligand gated sodium channel in the cell, so we'll write L for ligand gated. And in this example, we'll also say there are some voltage gated sodium channels near the voltmeters, so V for voltage gated. We'll also say there is a voltage sensitive potassium channel. Let's assume that the sodium channel is open when the voltage inside the cell reaches -40 millivolts. So again, at this point here. Let's also assume they close at +50 millivolts, so about right here, and that the potassium channel opens at +50 millivolts. Again, let's say a neurotransmitter binds the ligand-gated sodium channel, and sodium begins to enter the cell. So sodium enters the cell. This inward current causes the other positive charges in the cell to repel each other, as we showed in the last example. And this occurs until eventually the voltage near the voltage-sensitive sodium channel, right here, reaches -40 millivolts. So I'll represent that on the graph here, showing that as sodium enters the cell, becomes more positive until -40 millivolts is reached. At this point, the voltage-sensitive sodium channel opens and allows more sodium to enter the cell. Because the red voltmeter is close to this voltage-sensitive sodium channel, it detects a sudden spike in depolarization, which we can see on the graph here. Once the intracellular voltage reaches +50 millivolts, so the first peak right here, this channel closes, so no more sodium can enter the cell. At the same time, the potassium channel opens, which allows potassium to move from the intracellular space into the extracellular space down its concentration gradient. So potassium begins to leave the cell. As positively charged potassium leaves the cell, the intracellular voltage begins to normalize. So this returns to normal. This is called repolarization. It's important to know that the potassium channels normally stay open for a short amount of time, even after the cell has reached the resting membrane potential, such that the graph will dip down a little bit like this. This segment is called hyperpolarization. This whole process I've just described is an action potential. On the previous example, the initial signal was detected by the red voltmeter, but only slightly detected by the blue voltmeter. However, in this case, the additional influx of sodium will cause the voltage near the blue voltmeter to reach threshold, or -40 millivolts again, which will cause additional sodium to enter the cell. So the blue voltmeter will also detect a spike. Draw that right here. This process of depolarization and repolarization is what allows electrical signals to be transmitted and is one way in which the body communicates with surrounding and distant tissues. So again, as these positive charges reach the blue voltmeter, this other voltage-gated channel is open and additional sodium enters the cell, causing the blue peak I've drawn here. And then the whole process starts over again, potassium voltage channels open, and repolarization occurs. As we can see, multiple voltage-gated channels are necessary throughout the length of the cell to propagate signals across long distances. Okay, with this in mind, let's do some questions. How would the administration of diazepam alter the resting membrane potential of neurons? Okay, before we get into the question, I'd like to briefly mention that so far we've talked about action potentials in general and have referenced neurons. However, action potentials also occur throughout other cell types, such as muscle cells. We'll talk in a lot more detail about cardiac myocyte action potentials in the chapter on cardiology. Okay, so this question is asking specifically about neurons. Let's draw this out to make things clear. This will represent the cell membrane, this will be the extracellular space, and this will be the intracellular space. Neurons have GABA A receptors, which I'll draw right here. These receptors facilitate the passage of chloride ions from the extracellular space into the intracellular space, like this. This occurs when GABA binds to the GABA A receptor. So we'll draw GABA. Diazepam is a specific type of benzodiazepine, and benzodiazepines are a specific drug class that can be used to treat seizures. Benzodiazepines bind to the GABA A receptor, which I'll draw here, B for benzodiazepine. When this occurs, it facilitates the binding of more GABA to the GABA A receptor. So this causes more GABA to bind. This in turn increases the permeability of the cell to chloride, so more chloride enters the cell. Because chloride is negatively charged, this causes the cell to hyperpolarize or become more negative. So we'll represent that here with negative signs. As a result, the neuron must receive a greater stimulus in order to reach threshold. In other words, diazepams cause the resting membrane potential of neurons to be more negative. In patients whose neurons are easily excitable, such as seizure patients, this can be especially helpful. Okay, let's do another question. How would exposure to ciguatoxin alter the resting membrane potential? Okay, for step one, you need to be familiar with a few types of seafood toxins, and ciguatoxin is one of them. Ciguatoxin is a neurotoxin that's produced by several types of reef fish, such as a snapper. Exposure to this toxin results in widespread depolarization and paralysis. Let's draw this out. So this will represent the cell membrane of a neuron. This will be the extracellular space, and this will be the intracellular space. This will represent a voltage-gated sodium channel. Ciguatoxin, draw that with a C here, promotes the influx of sodium into neurons, which causes the cell to depolarize. So it binds to this receptor, causing sodium influx, resulting in depolarization. Normally, this results in an action potential, and then the sodium channel closes. However, in this case, the action potential occurs, but afterwards, the sodium channel cannot close. This means the cells cannot respond to additional action potentials, so they become paralyzed, which is why patients who are exposed to ciguatoxin develop paralysis. Okay, let's do one more question. The nerve tissue of a mouse is found to have a resting membrane potential of negative 70 millivolts. The equilibrium potential for sodium is positive 60 millivolts, and is positive 125 millivolts for calcium. Where will these ions move after opening their associated channels? Okay, in order to get this question right, you need to recall which ions are primarily concentrated in the extracellular space and which ones are primarily concentrated in the intracellular space. So chloride, sodium, and calcium are all ions which are primarily concentrated in the extracellular space. So chloride, sodium, and calcium are found in the extracellular space, whereas potassium is primarily concentrated in the intracellular space. In this case, we're asked specifically about sodium and calcium. Because sodium and calcium are highly concentrated in the extracellular space, they will naturally flow down their concentration gradient into the cell until their equilibrium potentials have been reached. So where will these ions move after opening their associated channels? They will both move from the extracellular space into the intracellular space. Let's draw this out. Okay, this will represent the cell membrane, the extracellular space, and the intracellular space. I'll draw two channels here, sodium and calcium. Okay, let's just focus on sodium for a second. When the sodium channel is open, it will naturally flow down the concentration gradient into the cell. As positively charged sodium ions enter the cell, the inside of the cell becomes more positive. Because the extracellular space has lost some positive charges, the extracellular space becomes more negative. This creates an electrical force that pushes sodium back and prevents it from entering the cell. So I present that right here with an electrical force arrow. As stated in the question stem, sodium has an equilibrium potential of positive 60 millivolts. So positive 60 millivolts. This means at positive 60 millivolts, the electrical force is strong enough to prevent the influx of additional sodium. So positive 60 millivolts. The same concept is true for calcium and other ions. In this case, calcium will enter the cell until the cell reaches positive 125 millivolts. At this point, the electrical force is strong enough to prevent the influx of additional calcium. So this force repels the influx of additional calcium. As you can see from the few questions we've done, it's most important to understand action potentials from a conceptual standpoint. You most likely won't be asked to plug numbers in on your Step 1 exam. As I mentioned earlier in this section, we'll go through action potentials in more detail in the chapter on cardiology. And that concludes this section.